Thanks, Martin. Thanks, everybody. Thank you for having me. Thank you for inviting me and bringing me all the way from New Brunswick. Just before I start, just a tiny bit about what I do in New Brunswick. I work at the New Brunswick Museum, where I'm the curator of Botany and Mycology. And this very nice picture looks so nice because it doesn't exist yet. This is the original building of the New Brunswick Museum from the 1930s. This is where we used to be. And this is the new building that they're currently building right now. And it should be open by 2028. So this is going to be the New Brunswick Museum moving forward. We're all very excited about the project and having a whole new building. And we're located in St. John, New Brunswick. Martin?
Can we see your office from the office window from there?
I hope they give me this one right here. That's what I hope. I don't think that's going to happen. But so we're in St. John, New Brunswick. So anytime after 2028, if you visit New Brunswick, stop in St. John. The museum will be open for everybody. And what I do at the museum, other than taking care of the collection for Botany and Mycology, doing outreach and coordinate things for the general public, my research is focused on mushroom diversity. So what you do, guys, here, but instead of here in New Brunswick, and a little bit by extension in all of Atlantic Canada, studying mushroom diversity there. And one of the ways how we do that, and you have heard a lot about sequencing in the last few days here, we have the MycoMap AC network, and AC stands for Atlantic Canada, where we're trying to sequence as many collections as we can from all the Atlantic provinces. And we opened the call for sequencing last year, and we already have over 3,000 collections sequenced from Atlantic Canada. And we hope to get many more this year if the forest fires ever stop and we can go back into the forest and get some more mushrooms. So that's basically what I do. But today I'm not talking about that. Today I'm talking about what is probably my favorite genus. I shouldn't pick favorites. They're all really great. They're all really cool. But some are extra cool. And I think Pluteus is one of them. Pluteus is a very distinct genus. It's a genus of gill mushrooms. And as Danny mentioned yesterday in his talk, is one of the more distinct groups because it has pink spores. And there are not many things with pink spores. And Pluteus is one of them. It has other morphological characteristics, but the pink spores and the free gills are two of the main things that are going to cut your attention from the first moment you see them. The other thing is that they often grow directly on wood or wood remnants. A few of them can grow directly on soil without connection to wood, but those are fewer species that do that. And overall, it has been estimated, traditionally, that Pluteus was a genus with about 200, maybe 300 species. Right now, we already have detected, just by using DNA data, 250, and we're sampling mostly Europe and North America, so we have the rest of the world to really sample in detail. So we estimate that probably we have at the very least 500 species of Pluteus worldwide. So quite diverse in terms of species numbers. One thing that I wanted to mention is that I'm going to be presenting a lot of results today, and I'm the one presenting them, but the work that I do is not possible without this list of people here. These are my collaborators that are mostly based in Europe, and these are my collaborators that are mostly based in North America. This includes both professional mycologists that work at universities, research institutes, but many of them are citizen scientists like you guys. They are part of mycological societies and collect mushrooms for fun and want to study them. Without these people that you see here, and many more that probably will get added as we get more collections, the work that I'm presenting is simply not possible. I would love to be all around the world, collecting all the time, but that's not how it happens. So getting other people to send me their collections is a big part of what I do.
Taylor's on there.
Taylor's on there, guilty. Dan is there also, yep. So first thing about Pluteus is that, as we already said, it's pretty easy to recognize when you find the mushroom in the field. But the next step to get closer to a full idea of the mushroom without even getting into DNA is put it under the microscope. Pluteus is a genus that to get a good handle on idea of the species, you need to put it under the microscope. And there are two main characters that are going to tell you which group of Pluteus you're looking at. And those characters are, how are the cells in the cap, what we call the pillae pellis. There is a group that have kind of elongated flat cells. There is another group that has round cells in the cap. And there is another group that has elongated cells, but they're standing upright. And then the other character that you need to look at is what type of specialized cells do they have in the gills? Do they have cells? These cells are called cystidia. They're basically specialized cells that surround the basidia that are producing the spores. So these cells might be, in this group, they're going to be thick wall, and they're going to have ornamentation. And in the other two groups, they're going to be thin wall and without ornamentation. So based on the combination of these characters, what type of cells do they have in the cap? What type of cells do they have in the gills? We have three sections. And sections are just subdivisions of genera that we use to group together species that are similar. Section Pluteus, section Celluloderma. Celluloderma means round cells in the cap. And section Hispidoderma. Hispid relates to hair because these upright cells kind of look like hairs that are on the surface. And you're going to see these names repeated over and over again, but basically it's just three main groups of Pluteus based on morphological characters. So I started doing, and when I did my PhD back in Spain, I was doing traditional taxonomy. I wasn't doing any DNA work at all. So it was all morphology. When I moved to the States for my postdoc, one of the first works that I did was looking at the taxonomy of Pluteus, but using DNA data. And you're going to see a lot of these phylogenetic trees in my presentation. I'm not going to go into any details about them. Just think, when you look at these trees, what this is trying to represent is how the organisms in the tree, each line here is a separate collection, how are these collections related to each other? So it's trying to answer two questions. Are these collections the same species or not? And how are the different species related to each other? Think of it as a kind of a family tree. So things that are closer in the tree are more related to each other, and things that are farther away in the tree are more distantly related. But again, I'm not going to go into any details about phylogeny. Just think of these trees as little family trees of Pluteus. So in the first phylogeny, what we found out, first of all, that classification that I just showed you into the different sections, pretty much it holds up with DNA data, which is good, because DNA tends to kind of change everything once you look at DNA. But for Pluteus, there is a couple of exceptions, but for the most part, that classification still holds up. We had almost 100 DNA sequences of the barcode, which at the time, almost 15 years ago, was a lot. My God, we had 100 Pluteus sequences. We were so happy about it. It was mostly European collections, a little bit from Eastern US, not so much from Western North America. And by that point, we said, well, we have at least 25 species or species groups. A species group or a species complex is what you cannot really tell how many species you have in that particular morphology. We said, well, we have about 25 based on the data that we had 15 years ago. But in many cases, we couldn't figure out exactly in each of these groups, well, how many species are really there? Is it all one? Is maybe this is one species and the next one is another species? And with the data that we had, it was really difficult to figure out exactly how many did we have. And it was also very difficult to figure out what very old North American names, the species that were described in the late 19th century, early 20th century, what did those names exactly meant? How could we apply those names to the collections that we see in the field today? So one of the first studies that we did after that first general one was to study the deer mushrooms. If there is a Pluteus that you're going to find in a popular field guide, it's probably going to be this guy. And deer mushroom, and the Latin name means exactly that. Cervinus is from cervus, which means deer in Latin. And it's mostly because of the color of the cap, but also people have made the connection that is because of the little ornamentations on the cystidia that look like little horns on the deer that I showed you before. So this deer mushroom, it has been reported from all over the world. But there have also been several species similar to the deer mushroom that have been described both in Europe and North America. So we decided to focus just on the species group around Pluteus cervinus and try to figure out, well, what species do we have in Europe? What species do we have in North America? Do we really have species that are in both places at the same time? And how did we do that? Well, this is kind of be the blueprint for all of the studies that I'm going to be talking about today. First, collaboration. As I said, it's not me doing all the work. Without collections and other taxonomies doing the work on their collections, also members of mycological societies, the general public, this type of a study is just not possible. At that point, we had over 300 collections just of the Cervinus group. We were just looking at this group of species. And we were trying to do what now we're calling integrative taxonomy. It's not that we're looking at the DNA and we hope that the DNA is going to give us all the answers. The DNA by itself tells us very little if we cannot connect it to something else. So we look at the morphology, the external morphology of the mushroom, what we usually do when we go out in the field. We also put it under the microscope, and Pluteus has a lot of characters under the microscope, so that's good. So we can use them to try to figure out the species. We look at the ecology. I think it was in Mike's presentation that he was asking me about a certain Pluteus, and the first thing that I asked him is, what was it growing on? Was it growing on hardwoods? Was it growing on conifers? Ecology is very, very important for Pluteus and for many other mushrooms. We're looking at the geographic origin of the collections that we were analyzing, and we knew the ITS, which is the barcode that we normally use, was not going to be enough. Just by itself, it does an okay job, but it doesn't give us the full resolution that we need. So we were using a second gene to do the molecular phylogeny.
What is ITS?
ITS is just one particular gene that for fungi is the one that we use to try to use it for species identification. So these were the results, and they were published in 2014. And we figured out that we had 26 species across the temperate areas of the northern hemisphere, and 12 of those species were described as new in the year 2014, not in the year 1905. In the year 2014, we're still describing 12 new species in one single paper. So there was a lot of diversity that without the help of DNA data was very difficult to detect. And if we just count the species in North America, just based on what was published in 2014, there were a total of 19 species of this group in North America. 10, which means more than half of the species that we have in North America, were not described until the year 2014. So that gives you an idea how much fungal taxonomy lags behind other fields like birds, plants, and other things like that. Now comes the moment of the truth, the first test of the evening. So you have seen a lot of mushrooms. I'm going to present you with, and I have to say, whoever fails the test has to leave. I'm sorry. You cannot stay for the rest of the foray. So just think very, very carefully about your answers. So these are 6 collections, and these are 6 actual collections that we use in our study. And you can see some morphology variation. What do you see that is different about the morphology? What characters do you see that are different in the different collections? Color, texture, okay? So different colors, we have white, we have different textures. So my question is, Do you think these 6 collections represent 1 species, 3 species, or 6 species? And I'm going to ask show of hands for this is all 1 species. 2, 3 takers, OK. This is 3 species. Yeah, a few of them. And this is 6 different species. So most of you were with the middle answer, which is the safer one, because you're not fully wrong or fully right. I see, OK. Well, second and final test of the evening. Another 6 collections. Here you don't see as much of morphological variation, I would say, but still you see some variation. And the question is the same. Do you think these 6 collections are all 1 species, 3 or 6? Takers for 1 species. Quite a few, OK. Takers for 3 species. You're becoming braver. I like it. This is 6 different species.
Wow.
OK? The answer is the first one is all variation within 1 species. This is all Pluteus cervinus. This is all the deer mushroom, all of it. The white form, the grayish form with very ornamented caps, the more typical brown forms. And these 6 collections that don't look that different from each other are actually 6 different species.
It's the habitat.
Part of it is the habitat. That's very good, Chips. I'm kind of tricking you because I'm not giving you the full information. I'm not telling you anything about the microscopy. I'm not telling you anything about the geography. I'm not telling you anything about the habitat. But you see the problem. The problem is if you're just looking at, yeah.
There's no problem. They all taste the same.
Yes, that's true. That's true. That's probably true. And the thing is, like, if you're just using morphology, it's going to be very hard for you to get to the right answer in terms of how many species do we have. So another thing that we found in this study is that in North America, we do have endemics. We do have species that are mostly in 1 geographic area. Oops. These are some of the species that are in the east that we describe as new. And these are some of the species that are only in Western North America. For some of them, we have learned actually that are not that exclusive. For example, this one, Pluteus elaphinus, that I described from Massachusetts, it was found, I think it was 1 or 2 years ago in Alaska, of all places. So it's still a North American endemic, but it's actually in both Eastern and Western North America. So a lot of regional species. In a certain region, you're going to have certain species that you're not going to have in other regions, which is an idea that for fungi was not that generalized for a long time. We thought that everything was basically everywhere. Another important thing is that most of the Western North American material that we use in this study was coming from projects like the one that you're trying to do here, projects that are led by citizen scientists, by regional diversity studies. They didn't come from academic projects or people at universities, even though some of the people were associated with those institutions. And this is one of those examples. This is Pluteus orestes. That at the time of the description, all the collections came from citizen scientist projects. They didn't come from my own collections or the collections of other people in California. And this species was described, the type collections is from Yosemite National Park in California. And this is what we know about Pluteus orestes today. Orestes, by the way, means mountaineer or hiker because it's a species that only appears in the mountain ranges of coastal Western North America, sometimes in the foothills, but basically all around the mountains. The original collections were mostly from here, California and Oregon. We know it goes as far south as the mountains around LA, as far north as Washington State. I assume it's probably in BC, in the coastal mountain ranges near near the ocean, but we haven't had any confirmed collections yet. And again, most of these collections, and these are the collections that are currently on iNaturalist, everybody can search for these species. These collections come from citizen scientists. These are not scientists working at research institutions trying to find the species. And this is for sure a Western North American endemic, and it's also is one of the few that has quite a few morphological characters that are distinct. But the work is not done. This was 2014, and we were very happy. OK, we're done with section Pluteus. We describe everything. Next. That wasn't true. Like just a few years later, Terry Clemens, which is another citizen scientist from Arizona, from the Arizona Mycological Society, she was finding these Pluteus, and she got them sequenced at the University of Arizona, and they ended up being a new undescribed species that we described just last year in 2024 as Pluteus arizonicus. And right after we described Pluteus arizonicus, people started to find the species all over the southwest of the United States. So yes, obviously it's in Arizona where it was described from, but it's all over the southwest, and it's even one collection from Southern California. All these dots that you see in the map are sequence collections that are mostly coming from the projects run at Mycota labs and other labs in the US. So again, participations for the projects like the one that you're doing here, those are the projects that are giving us the data that we need to understand fungal diversity in North America. It's not going to come from anywhere else. I can tell you in academia there are less and less people doing what I do, less taxonomies, less people who do taxonomy and biodiversity. So all the data is going to come from projects like the one that you're doing here. And there is still more to do. So even though we were very happy that we have figured out section Pluteus, we actually have even more species. We have these species that also Western North America from California to BC. This one that we already knew was there, also from California to BC. And this one also from California to BC. We have at least 3 more species to describe in this group that whenever I have time and I can sit down and do my homework, will probably see the light of day, hopefully very soon. And you see these labels? This is from the California Sequencing Project that, again, is mostly run by nonprofits and citizen scientists. So section Pluteus, OK, we still have work to do, but we're OK. We described quite a few species. We have figured out most of them. What about the other 2? What about the other 2 groups of Pluteus that I told you about? How many species do they have? How well do we know them? Again, we're going to do the same thing. Since it worked that well the first time, it's going to be based on collaboration. We need input from the sequencing labs, from citizen scientists, mycological societies. We're going to depend on having collections. Without collections, those are the building blocks that allow you to do everything else. Without the collection that you keep at a foray or where you are collecting, you're not going to be able to do this project. And for these 2 groups, we have right now over 2,000 collections that we have studied. And again, we're going to look at morphology, macro and micromorphology, ecology, geography, and 2 different genes to have as much genetic information as we can. And we come to kind of the central contention point in North America is how do we use the names of a species that were described over 100 years ago? Most of the species of Pluteus in North America that were originally described from North America were described by these 3 classic mycologists. William Murrill, which was the curator of the New York Botanical Garden and later moved to Florida. Charles Peck, who was the state mycologist. Imagine having a state mycologist. How cool is that? Yeah. Imagine that every state and province has a mycologist. Charles Peck was the state mycologist for New York, and George Atkinson that also describes some species of Pluteus. And this is some of the examples, some of the remaining collections from these people that are still preserved at those institutions. So how do we connect the dots? How do we take the collections from 100, 120 years ago and try to connect what we are seeing in the DNA analysis that we are doing? So the original collections, as I told you, they're preserved at the New York Botanical Garden, New York State, and other institutions. And those collections, over the years, they have been revised by people trying to understand the taxonomy of Pluteus. The problem is the conclusions from those revisions, sometimes different people will study the same collection and will report slightly different data. So you're like, what? What are you saying? You're supposedly studying the same collections. How can you be reporting these different data? So I was able to study myself most of the collections. So now I have my own original notes on the collections. But the main problem about these species is that even though more than 100 years have passed since these species were described, for most of them, there are very few, and for many of them, 0 new collections made in a period of over 100 years.
Wow.
So you are left with a description from 100 years ago, the original collections that were used to describe the species, and that's it. You're on your own. With that, you have to try to figure out what those names mean. I'm going to come back to this at the very end of the talk because we have some good and exciting news about these old collections. So for section Celluloderma, this is what the DNA data looks like, more or less. And each kind of big color blob is one species group that we're going to study separately. And for the other group, for the Hispidodermas, we have 3 big species groups. And for section Celluloderma alone, we have over 100 species worldwide. So very, very diverse, probably the more diverse part of the genus. And also, as you can guess, the more complicated to study and the one with the more difficult taxonomy. So this is one of the studies that was just published just January this year. And in this case, we were studying the Pluteus leoninus clade. Leoninus makes reference to lion because they have these very bright yellow colors. That's how the species was named. And in this study that we just published, again, just at the beginning of this year, we were trying to figure out, first of all, what is Pluteus leoninus? Pluteus leoninus is a species that was described from Central Europe in the 1700s. There are several species from Europe that some people say, well, they're all the same. Some people say they're different. But I'm not going to get into the European species at all. And what we have in North America is one name by Atkinson, one of the early mycologists that I mentioned later, that he described from New York, and he called Pluteus flavofuliginus. That we were not sure, is this the same as Pluteus leoninus from Europe? Is this different? And all of the species in this group, well, not all, but most of them have these bright yellow colors. The cells on the cap, they stand upright. And if you look at them fresh, they will have that yellow pigment. And the cystidia, the cells in the gills, have these little kind of finger-like structures on the top. So one thing that we were able to do, luckily, is we were able to actually sequence the original collection of Pluteus flavofuliginus. And it's right here with all of the Eastern North American collections. So this is Pluteus flavofuliginus here. And this is Pluteus leoninus from Europe and Asia. So based on DNA data, there are some differences, not a lot, but quite constant considering the geographic origin of the collection. So Pluteus flavofuliginus is one of the older names that we can bring back to life. We don't have to describe it as a new species. We already have a name for it that we were not sure if it was actually right or not. And even more exciting, out west, we don't have lions, we have pumas. We have the Western North American version of Pluteus leoninus. This is Pluteus pumae that we describe in the paper just this year. And you can see how the sequence collections group very nicely together all the Western North American collections. Pluteus pumae is now from California all the way to BC. And I'm pretty sure you're going to find it here in Alberta. So this is the current distribution. The BC collections are not on iNaturalist, but we have some. And it's also at least all the way to Colorado in the US. In this paper, we described 4 additional species from North America, all part of the same group. And these 2 that I highlight here, Pluteus esperius. Esperius means western because it's in western North America. And Pluteus insularis because the type collection comes from Anticosti Island off the coast of Quebec. These 2, the type collections, they're both Canadian collections. This one is the type of Pluteus esperius collected in BC by Oluna Cheska. And this is Pluteus insularis collected in Anticosti from René Leboeuf. And this is how a modern collection, a modern description of a species looks like. You have representative collections showing morphological variability, and then you have all the microscopic characters of the species. So lots of new species from North America. And one of the things that we were kind of going back and forth is like, well, again, there are all these older names from these older mycologies that we're trying to bring back to life. Is it possible that any of these species actually has an older name available and we haven't figured it out yet? So in this case, we were able to figure out that 4 of the species that were described by Muriel from the North American Northeast, mostly from Maine, New York, Connecticut, these 4 species are actually synonyms of Pluteus flavofuligineus, which is the species that is present in that area. We do not have yet molecular data for these types, but just based on morphology and the geographic origin, we're pretty confident that these are just synonyms of Pluteus flavofuligineus, which is great because at least now we know what those names mean, and we don't have to worry anymore about them. And that was for section Hispidoderma. And for the last group, for section Celluloderma, we have about 160 species currently sequenced. Again, each one of these color blocks is a whole world of project and collections and collaboration. And I'm going to tell you just a tiny bit about the 2 that are already published and how it's important for North American diversity. This is the Pluteus romelii group that their revision was published 3 years ago, and this is the Podospilius group. And for these guys, for the romelii group, we have 4 species in North America. And before our study, we were using just one name for this whole group. Oh, this is Pluteus romelii. This is Pluteus romelii. Well, Pluteus romelii is here, happy, living in Europe, and these are our North American species that are present here. So one of the names that we have is not a new species, but a species by Muriel. Muriel in 1917 described Pluteus fulvivarius from the coast of Oregon. And while we have not been able to sequence the type yet, we have been able to study morphologically. And we know that we have modern collections that correspond to that species. So this is Pluteus fulvivarius. The yellow stems that you see here are kind of the tell character for the Pluteus romelii group, for the whole group. For this particular species, this one is relatively big compared to other species in this group. And the gills, they mostly stay yellow up until, because the spores are pink, so they stay yellow up until there are so many spores on the gills that the gills eventually turn pink. So again, one species that we were able to bring back to life, even though that hasn't been used. Chips.
Is it normal for the Pluteus to be found on a harvested tree or the parts of a harvested tree?
Yes and no. It's not an uncommon habitat like wood chips or pieces of wood that are just falling on the ground. Most of the Pluteus would prefer like really well-decayed wood in the forest floor. That's their preferred habitat. But the habitat that you see here, it's also a possibility for Pluteus. Yeah. These are other 2 species from eastern North America. Austrofulvus means the yellow one from the south because it has a much southern distribution than the other one that I just showed you. Parvisporus, parvi means small because it has the smallest spores of the species in this group. This is Pluteus vellingae, which was dedicated to Elsa Vellinga, which is one of the more active, both professional and amateur at the same time mycologists in North America, even though she's originally from the Netherlands, and she has done quite a bit of work in Pluteus. So this, and she actually collected the type collection quite a few years ago. So it was dedicated to her. And again, these yellow colors on the stem, often on the gills, the brown caps, that is typical of the whole group. Then you either have to go to DNA or to do a very detailed microscopy analysis to be able to figure out exactly what species do you have. The other study that we have already published is the Pluteus podospilius. Again, podospilius is a European species. Here in North America, there were 2 names. Remember this number, 2. 2 names for the species in this group. Pluteus seticeps, which was originally described from North America, and Pluteus seticeps variety cystidiosus. Those were the 2 names before we did this revision that were used for basically all the collections of the species of this group in North America. How many species do you think we have in North America before I go to the next slide? Numbers?
More than 2?
More than 2, yeah. More than 5? 10. You're almost right on the money. First, this is Pluteus seticeps, and this is Pluteus cystidiosus. So these are not even varieties of the same species. These are 2 completely separate species. So that was one of the first things that we figured out. The other thing that we figured out is that we have 10 species in North America. From 2 names that we were using, all of a sudden we have 10 of them, of the species in this group. So it's quite a jump. And again, a lot of the times, if you put enough effort and try to figure out what those older names mean, you can bring those names back to life. For many years, Pluteus seticeps was considered a synonym of Pluteus podospilius. Let me go back to the phylogeny. Pluteus podospilius is right here in the DNA analysis. Pluteus seticeps is right here. So they're clearly not the same thing. So again, we brought back to life Pluteus seticeps. This was a species from Atkinson. And we had another 5 species present in North America that had names already available for them.
Who gave them those names?
The authors that you see after the name, that's the abbreviation for the author. And the ones that say SP Nov, that means new species. We gave them the name.
Oh.
We described it.
One is abscon, one is inexpectat.
Yeah,
So,
those are the best names that we can figure out for the species. Sometimes you try to figure out a name that means something related to the species.
what does that mean in Latin?
Absconditus means hidden, because without DNA data, it was very difficult to figure out that it was there. Inexpectatus means unexpected,
Unexpected.
because we figured out that species was there about a month before we were ready to send the paper for publication. So we were like, not now, please no. So yeah, we always, eventually you run out of names that mean something, but we try to give them names that have some meaning. So 6 of the species that we presented in the paper, they were either described as new or they had older names available for them. These are some of the new ones. This is the Inexpectatus one. And this was found by René Leboeuf in Quebec, in 2 collections actually pretty close to where she lives. She didn't even have to travel far to find a new species. So in North America, it's very true that we have undescribed species literally in our backyards. And we have 6 with names, but in the same study, we had 4 species that, based on DNA data, we knew that they were different from everything else, but we couldn't describe because we didn't have enough collections. This one was only known from 1 collection in Ohio. We have these environmental sequences from Mexico. We have this collection from Mississippi, this collection from Indiana. So we only had one, so it was very difficult for us to formally describe them, but we know they're there. So at some point, we're going to be able to find more collections. Actually, just this week, somebody found the second collection of this guy here. So we're probably going to be able to describe it pretty soon. So that was in the paper. 10 species, 6 with names, 4 without names, because we didn't have enough material. As soon as we published the paper, there was another one that we missed. There was another one that we couldn't put in the paper. And this is from the Pacific Northwest, so most of the collections come from Danny and from other people that are doing sequencing in Western North American collections. Right now, we only know it with a temporary code because we have to name it something until it gets a formal name. So it's Pluteus Pacific Northwest number 13, but we hope to formally describe it and it will have a formal name pretty soon. And again, I know I'm repeating myself, but this is all people like you collecting and keeping those collections, sending them for DNA sequencing. Otherwise, this type of discoveries will simply not be possible. And is these projects, the projects done by Mycology labs, by Ohio Sequencing Lab, by Danny in the Pacific Northwest, we now have over 1,500 sequence collections of Pluteus from North America. That's a lot of homework for somebody. I don't know who is gonna be looking through all those data, but that's a lot of data to go through. We'll still have a lot of things to discover and to formally describe. So I'm almost done. I just want to tell you that today I told you about section Pluteus, which had a whole study just for the whole section. We know we still have some loose ends to tie up. In section Celluloderma, we published 2 papers for this blob over here and this blob over here. We're still working on the rest. In section Hispidoderma, we published this yellow blob over here. We're still working on the rest. Yep, Chips.
The governments are spending money on artificial intelligence. Can't that solve all our problems?
I don't think so. I don't think so, because then, well, first of all, it will be so boring to have artificial intelligence identify mushrooms for you. What do they know about identifying mushrooms? Maybe to some of the genetic data, AI is going to have more applications. For taxonomy, I think we humans will still like to be able to identify the mushrooms that we find.
Excuse me,
Yeah.
could you just go back one?
Yeah. This. Yeah. Yeah.
Because you know what? That's Saskatchewan and ours.
Oh, great, great. So the last thing that I wanted to tell you about, and this I'm trying to kind of do kind of full circle moment and going back to some of the things that I told you at the beginning. One of the groups that is by far going to be the most complicated is all the small brown Pluteus. It's what we call the Pluteus nanus group. And these are all small, and the caps are usually brown. Some of them are gray or have some olive color to them. In the phylogenies, they're not even one single group. They are in this green clade here, in this one here, but morphologically they're very similar, so we're gonna study them all together. Just this group of small brown mushrooms, and everybody knows that small brown mushrooms are everybody's favorite. When you're out in the field, everybody collects all the small brown mushrooms. Just this group. We have 97 species just of these small brown mushrooms. Of these 97, well, 15 of them are from the Southern Hemisphere, so that's a battle for another time, maybe another life. But in the temperate Northern Hemisphere, we have 82. I'm including the Caribbean because a lot of them are kind of in both places. Only 30 of these 82 species have names. The rest, 52, no names. They're most likely undescribed. For a few of them, we might be able to bring back to life some of the older names. And I'm not going to go in detail at all in the tree, but basically the things that are marked with green are things with names. Things that are marked with blue are probably undescribed species from Europe, which should be the place that has had more studies and has everything described. No, there are still plenty of undescribed species there. Things in purple are from undescribed or likely undescribed from Eastern North America. Things in yellow, undescribed from Western North America.
And they're all edible.
Sure, at least once. And the last example that I'm going to give you is this guy. This is Pluteus olivaceus. And olivaceus means with olive color. You can see some of the greenish, gray-green colors in the cap. And this is a European species. And we have quite a few collections from Europe. We were able to sequence the type collection, the original collection. We are sure that's the name for this group of collections. We're pretty sure. But you see there are two other things around this. One is this Pluteus affinis olivaceus, and then this that I have here as CF californicus. CF means, well, we have to double check that that's actually the one. And californicus is one of those old names that we are trying to bring back to life. And as I show you in most of the other studies, it's very difficult to sequence the type collections. These are very old collections, 125 and 150 years old. Pluteus californicus was described from Pasadena, from California, by Alfred Mycology, which is, he worked most of his life in California and Arizona, but actually he was born in Quebec. So go Canada. And the original collections are preserved in the New York Botanical Garden. And this is what these original descriptions look like. 4 or 5 lines with very general characters and the original collections, and that's all you have. And with that, you try to do your best and figure out what this means. Unless, now that we have nanopore sequencing, we go back to these type collections that we tried to sequence in the past, and we were not able to, and now we try to sequence all the type collections that we have. So we made a trial run, not with all of them, just with 20 collections. We selected 8 types from Muriel, from the New York Botanical Garden, and the original collections for 3 species from Peck, from the New York State Museum. So 20 collections in total. And I said, this is not going to work. I have tried to sequence them in the past. It's not going to work. They're too old. It's not going to work. We sent the 20 collections to Mycota, and 18 out of the 20 work in the first try. We now have DNA sequences for many of these original collections, and now we don't have to guess anymore. These are the original collections of Pluteus californicus, which match very nicely with our modern collections of Pluteus californicus that we have sequenced already. So now we don't have to guess anymore. Now that we have access to this new sequence technology, we can sequence those original collections and bring these North American names back to life and know exactly what they mean. This is Pluteus californicus. It's similar to the European olivaceus in that it has these greenish colors on the cap, especially when they're young, and is a species that right now is only known mostly from Southern California. We do have a collection from San Francisco that is not here on iNaturalist, but basically coastal California, and this is the only place in the world where this collection, this species apparently grows. And the last thing is another Pluteus that is marked here in yellow, and as I told you, yellow are the species from Western North America that have not been described yet. And this collection comes from a place that might be familiar to some of you. Calgary, what is that? Calgary. I don't know, what is that? What? Who is this guy? Taylor? What? This is a collection from Taylor, from Calgary, that we have sequenced. It's in the general group of Pluteus olivaceus and Pluteus californicus, but it doesn't correspond to either of them. So this is probably a new undescribed species that you have here.
Very, very cool. So because it's not from California, would you just cut the Cali off and call it Pluteus fornicus?
Yes, perfect, perfect. Done. We don't have to think anymore. We have it. And you have all the documentation, the great documentation that Taylor did on his collection. We have the DNA data. I think for this one, we just hope to find maybe another one sometime, and we can. So if you see a small Pluteus, even if it's a small, boring-looking thing, it's not going to be boring. It's going to be very interesting. At least there's going to be one person, me, that is going to be interested in that collection. It's going to be something that is going to be cool and worth studying. So again, keep doing what you're doing. You're doing great projects everywhere, and thanks to all the collections that citizen scientists across North America are sequencing, we're finally going to be able to get an idea of the species that we have across the continent. So thanks again to all the people who have contributed collections to all the places where the DNA sequencing is being done. And with that, that's all that I wanted to tell you, and I'm happy to answer any questions. Yeah.
What is the criteria for the clade?
The criteria, basically the clades, you don't decide how the clades are defined. Like when you have your DNA sequences, there is a program that is going to organize those DNA sequences, and there's another software or program that is going to analyze them. And the end result is a phylogenetic tree like this. Basically, this is going to tell you all these sequences here from this branch are more similar to each other than to this other sequence that is in a separate branch. And these two are different from this other one here. That is the general idea. Then you, as a scientist, has to decide that these particular three branches are the ones that are defining groups that should be recognized as a species. Since all of these are from Europe and share the same DNA sequence, all of these are from California and the West Coast, and this one is from Alberta, and all three groups have different DNA, I think most likely these are different species. But that's the scientist doing the analysis of. Okay. The program only gives you an output, a phylogenetic tree. The tree itself is not the answer. You have to, again, put together all the information. What does the DNA variation mean? What does the morphological variation mean? What does the geographic variation mean? And try to make sense of all of it. That's kind of where the difficulty comes in. How do you put all the pieces together? Yeah. Danny.
To what do you attribute the nanopores successfully sequencing such old collections where you tried, presumably with more sort of focus and individual attention before and that had failed?
Probably just better suited to deal with more degraded DNA. I think it's a matter of how degraded the DNA is in those collections. I don't think it's a matter of quantity of DNA. I think it's more a matter of nanopore and also Illumina. Even some of the European types, they have been sequenced with a different sequencing technology. They're just better suited for more degraded DNA than traditional sequencing is. It's completely true what I told you. When I sent those 20 collections, I was 90% sure these are not going to work. I have tried them before. They never work. And when basically all of them work on the first try, I was like, great. Now I'm going to sequence all the types for which I have already made an interpretation to see how wrong I was interpreting the old names. But I hopefully going to be able to sequence all of them, and relatively soon. So yeah, it's very exciting because you don't have to guess anymore. And you're able to bring names that people doing the work, 100, if we think taxonomy is difficult now, doing taxonomy 100, 125 years ago, and they got a lot of things right. Obviously, they got some things wrong, but they got a lot of things right. So being able to bring those names back to life is great. Yeah. More questions?
Where can I get a T-shirt like that?
On the internet. Everything is on the internet. Yeah.
What is Pluteus again in Latin? What does it mean?
It means shed or shield, like a protective structure. Like a barrier, or yeah, a shield, a protection, yeah.
Why do you think this genus was named Pluteus?
I don't know why. I think probably because of the shape of the cap. It tends to be like really flat, like something you would put in a structure to protect it. I think that's probably the reason why.
So we couldn't solve here the question. What was the question?
The question is, what does Pluteus mean? What is the etymology of the name? Yeah.
So is that about 1 centimeter across, or
The
the mushroom?
the mushroom is pretty small. I think it's probably like 2 centimeters.
Okay.
Yeah, yeah. They're tiny. The ones in this group, they're really tiny. Okay?
Its little shield.
They're the little shield, yeah. I'm around tomorrow, so if you have any more questions, anytime, come see me. Happy to talk to you about Pluteus or anything mushroom related. Thanks.